I have the passport inside it.
Yes, passport is the most.
Oh man, this doesn't come up on the screen. Okay. Useless. No, it worked, but that does not like. It's okay. I'm here. Oh wow. There are 2 chairs here. All of you PhD students? Mix. Okay. Amazing. Amazing.
So I just introduce Mohammed Alser today. He's had very convoluted tweet, so Mohammed attended his PhD in computer science in August 2018 from the University in Turkey, and then afterwards joined PhD. I did a research group where he eventually became a lecturer and a senior researcher, and was leading the informatics research in the same group and got courses in the totally of bioinformatics. Today he's a senior track as a lecturer at bioinformatics and computer science, the Department of Computer Science of
Thank you so much, Gia. It's a great pleasure being here, Barcelona. This is my first time here, and so far it looks amazing, at least the weather. We'll see tomorrow the city. So yeah, so it's amazing. We will see the story behind the title, but for now, the title, there is plenty of room in the data itself, and I'm going to talk about this for the next 1 hour. I would like to thank first Adria, Jesus, and Robin. Where's Robin? The big guy for today. We'll have big event, hopefully. And I guess Mikol. Oh, I was looking for you. Good morning. Yeah, Santiago. So it's a great, really, to see everyone today. And thank you so much for inviting me. The big news for today, I'm hiring PhD students. I'm really looking for outstanding, strong student in software optimization, hardware design, processing, and memory. So feel free to share it among your peers. And this is our link to our website. So you'll see different opportunities. Sometimes we host self-sponsored students as well, and so on. All right, let's get this closer. If you look at this picture, we see different people with different traits, different phenotypes. Could be body mass, the height, skin color, hair color. And we were wondering like what caused all these differences. And when we look deeper, we will find what we call as our human cells. And in these cells, we got what we call as a DNA. The DNA is basically the set of genetic instruction. You can think about it as a recipe to make a food. And whenever you turn or enable any of this instruction, you cause something in the body. It could be as a phenotype, or it could pass through the generation, to the next generation, and so on. The DNA exists in every human body cell except the blood cells. We don't have DNA over there. And since it's the instruction that codes those phenotypes, we will always wonder how to read those instructions. Knowing those instructions will enable us to know what causes us to be healthy or unhealthy. So ideally, you would like to get the genetic sample and get the full text of our DNA. And by reading the DNA, we'll be able to know those instructions. And if you look deeper to the DNA, you'll see that it's composed of 4 molecules or 4 alphabets. Those adenine, guanine, cytosine, and thymine. So those are chemical molecules, basically. And knowing the whole set of those chemical molecules will tell you this instruction. Unfortunately, since 1977, which is the first time we tried to read a virus genome, until today, we cannot read the full genome as one piece. What we have is a very laborious process. We get the genetic sample, and then there is a technician sitting in the lab with a white coat, try to prepare all those molecules. So try to isolate the plasma, the red cells, and so on, leaving only the DNA over there. And then he will try or she will try to chop the DNA to break it down into smaller pieces. So who's breaking down the DNA is ourselves, okay? So we got those small segments out of the DNA, and then we pass it to what we call as a sequencing machine. Sequencing machine will try to read these from chemical molecules into something either as images, raw signal, could be amplitude, electrical values, or it could be a video. Okay? So those what we call them as raw sequencing data. So we convert the chemical molecules into computer science data, basically. Now we convert each of those raw sequencing data into what we call as a read. So each of those pieces, which correspond to the chemical molecules content of each of those in the segment, we call it as a read. And those reads will start processing them using computer algorithms. So everything we saw on the top is processed in the wet lab. So there is a technician doing that, not us as a computer scientist. Everything you see at the bottom could be done in the dry lab using computer, starting from the raw sequencing data all the way to the analysis to infer the disease and so on. All right, so this is a picture of recent. Those are very modern. This, for example, just 2 years old. This is 1 year old. This is probably 2 or 1.5 year old. So those are 3 prominent sequencing technologies: Illumina, Oxford Nanopore, and PacBio. This could be fridge size. This is also fridge size. This is really handheld at the palm of the hand. And this is the modern device of the version of MinION. And you can see they are different in read length. So this is very short. This is the ultra long read, but the range is really wide. So it could be from short, 100, similar to this, all the way to 2.4 million long each piece. This one normally 20,000 fixed length, or sometimes from 10,000 to 30,000. But also they are different in throughput and price and so on. For example, this guy, let's focus on it. This is the older version of it, but still same principle. This is what we call as a flow cell. And this is the device, the sequencer itself. This thing costs alone about $1,000. And once we do the sequencing, we throw it away. It's no longer reusable. So you pay $1,000 and then done. But this one you keep. It costs about only $2,000 to $3,000. This is in the US. I don't know by importing, exporting, how much it will cost. But if we connect it to USB device to the computer, we can start seeing the raw sequencing data going there. And then we need to convert them inside the computer into those reads. Now, you can imagine, you can think about those individual reads as the puzzle pieces. And it is. Why? Because once we got those puzzle pieces, we had no idea about how they are interconnected, which chromosome they came from, which individual they came from, and so on. So we need what we call as a reference picture. So we try to build those individual pieces and reconstruct the genome again. So remember, we break it down ourselves into pieces, individual pieces, disconnected, and then we want to connect them back. Now, why we do that cycle, why we break them down, and then we need to reconstruct them again, this is related to the chemistry. So we have something we call enzyme, and this enzyme, it's like protein, and they are responsible on reading each of those pieces. So those enzymes have limited lifetime. So you might initiate them, they start reading, but then they degrade, then they die after some time. So to exploit high throughput out of all those pieces, We need to really break it down into smaller pieces and then start sequencing those individually. Remember, each of those pieces is only ACGT data. There is no information about how they are connected together. Now, this looks like easy, but in this scenario, it's a bit more difficult. In this scenario, we don't have reference genome. We don't have the reference picture to look at. In this case, this is the same individual. Both of them belong to the same human. But this one could be from cancerous cells, like we extract a genomic sample from the cancer. This one from healthy cells. And then what we got, individual reads. And now we want to compare the reads together to build what caused the cancer. Okay, we don't have a reference genome here. So this is a bit more complex than the previous scenario. In the third case, we don't know who is donating the genomic sample. So imagine we just pick a sample from here, from the table. We might get a lot of viruses, a lot of bacteria, and so on. So in this case, we cannot compare to a single genome, and we have to get a database of whole known genomes in order to reconstruct the guys in those pieces. So we keep comparing every piece to all those individual in the database, and then we try to guess, oh, it's coming from this virus, but not this bacteria, and so on. So this is a very complex step called metagenomic analysis, the previous one called genomics analysis, and so on. And there is a lot of scenarios how we can use those pieces coming from the sequencing machine. All right, now if we look at the computer science side, so we try to read those genomes. Think about this as sequencing machine side. So we could do 48 genomes in 2 days to read them all in parallel with high throughput. But when we go to the analysis side, we see it's bottleneck in the computational side. We can process only single genome in about 32 CPU hours. So there is really significant barriers to efficient analysis. And this is our goal, how to improve the computational side of this whole process. Now, the first thing, if we look, those machines are really dedicated, specialized machine with high throughput. They are able to give you a huge amount of data. But the analysis is always done at general purpose computers, right? They're not good at one thing, they are good in everything. And that's the first barrier that we had in today's genomic analysis. Now, the other problem is that we are doing the analysis really far from where we generate the data. We generate them in a machine, and then we transfer it to the computational center in hospital, for example, in order to do the analysis. And if we look at it, if we try to transmit this wirelessly, for example, 1 bit is 1,000 times more expensive than processing 32 bits at computation in today's computers. So this is extremely expensive in terms of energy to transfer 1 bit. Think about the huge high throughput. We think about it in our machine. So we would like to transmit all of this data over wireless communication, then we'll pay a huge cost for that. But that's not the only problem. Whenever we talk about wireless communication, we always talk about hacking those data. And remember, this data is not something special to you only. This can reveal your mother, your father, your sibling DNA. And by revealing the DNA, you could get blocked from health insurance, increasing the prices. Hospital could block you from getting treatment, especially in the US, and so on. And there is a lot of true stories about this. Now, if we want to move from, instead of transmitting wirelessly those data, want to process it locally. So we have to move the data from the sequencing machine to the SSD, all the way to the DRAM, to the cache hierarchy, all the way to the CPU to get processed. However, this is not better than the previous scenario. Let's see. First, we have the problem of data locality. So we are moving the data. We might need it later on, but because of the limited space, we are flushing it out. So we are moving a lot of data through this strip, and then we have only 1 kilobyte of the CPU. We process, we need this data later on, but we have no choice except just flushing it, move new data, and so on. And the DRAM today, these days, is really huge capacity, like 1 million times larger than what we have at the register file. But also, if you want to move data from the DRAM, not from the SSD, from the DRAM, a bit closer to the CPU, you have to pay about 400 times more expensive in terms of the latency to move this data to the CPU. But also we are going to pay power because it's about 100 times more expensive to move the power through the buses, which are power-hungry buses connecting all those components together. All those comparison to the CPU side. Now, everything we did in this trip, we call it as data movement, and this is extremely expensive, and sometimes it costs about 60% of the total system energy. So we are not talking about computation yet. So think about any software you're running in the CPU, 60% of it will be just for the data movement. Now, when we look at this figure, we'll see the processor performance is shooting up, is going to improve over time, which is something great. But when we compare it to the memory performance or the bandwidth, it's not getting as better as what we have in the processor. So we always have what we call as memory wall, which is the gap between the processing improvement and the memory improvement. And we see there's a huge memory wall or gap between those two elements. And that will tell you the importance of how to have efficient data and reduce that amount so that you reduce the number of trips going back and forth to the DRAM. With these years, focusing on AI, you'll see this wall still exists. This is the compute power of the GPU or of the transformer size. And you see that 240x every 2 years, doubling in terms of the parameter size. But when you look at the memory, it's only doubling every 2 years. So the memory wall is also there with today AI. And actually, it's getting worse with the memory-intensive application as an AI, for example. All right, now to solve all those issues combined, we need to look at something we call the compute stack. The compute stack. And when I build this slide, there are two papers that we always like about this topic. The first one says there's a plenty of room at the top. It means at the top of the compute stack, meaning that we need to improve the software behind those computation. But also there is a very old talking about there is a plenty of room at the bottom. And the bottom of the compute stack means the hardware. So we need to improve the hardware side. We need to improve the software. But there is a topic that's usually ignored in this area, in genomics, which is the data itself. The data is a huge problem in genomics, and nobody touching it. There is a huge need for improving the genomic data itself. And that is the story behind why there is a plenty of room for improving the data or for improving the genomic analyses by improving the data itself. Now, the first issue we have that all genomic data optimized for human. They are optimized for our naked eye to read them. And I don't know what's the purpose of that. So the message we have in this talk is that we must shift the paradigm from human-readable format to performance-aware genomic data. Whatever benefit the computer, we should go for that right away. There is really no need to open the file yourself. We are talking about here 70 gigabyte or 1 terabyte of data. There is really no need to open it and start reading it line by line. So let's see the first example, what we call as performance-aware data representation. Remember, we have 3 prominent sequencing technologies, those different companies, different output, and so on. But the nice thing that we do abstraction to those different formats, and we convert all of them into what we call as a FASTQ format. Okay, so it's no longer images or amplitude values or video, but we have a text over here, so you can really open it and read it. Now, those FASTQ files, they are publicly available. So as a researcher, you don't need to go through the sequencing step. So you can go to SRA database in the US. You can download any FASTQ file you have. Or you can go to the European database. We call it ENA. So you can download. And there is a huge overlap between the two databases. So you can find the same sample in both of them. Because open to public, so anyone can upload their data. Now, the FASTQ format itself, it's something really old. So it's introduced in the late 1990s, and it was used by the Sanger in the UK Institute. They were having the FASTA format in that day. The FASTA format just did the genomic sequence itself. But once they start using the sequencing machine, they saw an error introduced by this machine. So they want to quantify that error and whether it's relevant to each character in that genomic sequence or not. And that was the purpose why they moved from FASTA to FASTQ, by just adding what we call as a basic quality line to those data. And this is the format. So every read, every picture you get from, let's say not picture, because the picture is not single read. It's basically each pixel in that image correspond to one character in the read. So the whole image correspond to, for example, let's say 100 by 100 pixel image. It corresponds to 100 by 100 character, or the first character from 100 by 100 reads. So it's not really one image per read. Okay? However, if you think about as a read-wise, so every read should have 4 lines in the FASTQ format. So whenever you open the file, you should interpret it as 4 read block or 4-line block. The first line is what we call the header, which has a unique identifier to identify that read later on. The second one is the read content itself, which is the ACGT data. So you have the chemical molecules over there. The third line, we call it a separator, but sometimes we have the header just duplicated over there. Again, there is no reason. It doesn't make any sense, but we do that. Okay? So we copy or replicate the header in the third line as well. Now, the fourth line, which makes the difference, the huge difference between the FASTQ and FASTA. So everything you see here, you can call it as FASTA file, except, of course, this letter need to change to greater than sign, and then you can call it FASTA format. But if you add this, then we can call it FASTQ file. Now, what is this line? This line is the ASCII character. Okay, so each character representing the corresponding quality of that character. Okay, so why we need ASCII character? Why not just numbers over there? Because we need really higher range than the numbers and then the English alphabets and so on. We need about 120 values to uniquely represent each of those qualities. And you can think about it going from the exclamation mark all the way to the this mark. And that would represent something like 33 in computer, because the first 32 characters are not visible, so you cannot see them in computers. So that's why we decide from 33 moving to about 120 or something like that. But today, in today's sequencing machine, we are not using that range fully. So we need just like 40, 50 values to represent any decent data. Okay. So now how we read those values. So if we see like C or 3, what does it mean in the quality? If you look at this range, we see here, this is the first character in that range. And it's going from 0 all the way, sometimes exceeding 80, sometimes 90. So you need to replace this value in this equation in order to get the probability of being erroneous or of being correct. Okay. So you just replace the Q here with this number, and then you'll get the value P. For example, let's see 20, which means 5. So if you see 5 in that file, it means 99% it's correct value. Okay? Who's producing this value is the sequencing machine itself. It's telling you, I'm confident by 99% that what I'm giving you is correct. If we move to 40, the accuracy starts increasing, 99.999%, and so on. There are a lot of issues with today's FASTQ file. One of them, why we replicate the header. This is the easiest. Doesn't make any sense. Okay. Second thing, why we need to split the ACGT content from the basic quality. Third thing, what if this basic quality starts with the @ sign? If it starts with the @ sign, then it's confuse computer algorithms to read those files, for example. And there are many, many problems in terms of computation. For example, when we talk about O and T, it's 2 million characters long, each of those pieces. So imagine you are reading a whole line that's 2 million long character. You cannot fit it in maybe in register file or caches and so on. So you need multiple trips to solve that issue. And plus, you need to visit this line and this line together because they are really correlated. So every character here is representing something related to that. So it's not like you need to load this line and then do computation, and then you need to load the other line, and so on. Especially when you talk about assembly, but not read mapping. From read mapping, sometimes we can ignore this line. We can use this line alone. Here's an example. I don't want to go through the list of issues in the previous slide, but I will show it visually here. This is an example of what you really will get from the machine. This is, again, 4 lines correspond to 1 read. Okay? This is the read itself. This is the header. And what you see from the header, this is Illumina machine. What you will see from the header, the first thing is telling you what machine you are using. So this is the name of the machine. And imagine if you have like 10 million reads in the file, that piece of information is really repeated all over those reads. So if you have like 10 million reads, you have 10 million words with the name of the machine. This is extremely bad. Now the others are like, for example, the flow cell that you are using, what kind of identifier for this machine, for the cable, for something like that. Okay, you can add many information that you might need to the header. Now, if you want to download the data from the SRA, you'll see something like this. So this is from the machine itself. Once you upload it to the SRA database to be publicly available to everyone, it will be converted to something like that. So the first change, you have the accession number over here. This is a unique identifier added by SRA database itself to your file. So it will append it here or to have it as a prefix before whatever line you have in the header over here. So this line will be copied in the middle. In the beginning, we will add what we call as sample ID or unique identifier to the sample. And then we have the read number. And then we add at the end the length of the genomic sequence. Okay? And then we duplicate this line to here. And then we have this as SRA format. Okay? Now, if we would like to analyze it by, for example, Minimap2. Minimap2 will ignore everything after the first space. So if you send this file to software to do the analysis, it will ignore everything after that space and just consider this part. So you cannot really reconstruct your FASTQ file from the output of Minimap2 or from the output of any of those analysis software. Now, why they do that? Just to optimize, to reduce the size of the output and so on. But those information are extremely important because it's telling you what machine, what technology you are using. And that is really beneficial for quality control, for assembly, for many other analyses that rely on the quality of the machine. So without knowing what range you are using here, what quality you're expecting, it's really hard to control the quality. And you need to do try and error every time you want to filter those kind of files. Okay? So by losing those data, you are losing a lot. Now, what we are proposing to solve all of those issues is to convert this data from FASTQ into what we call as FASTA. In the FASTA, we are introducing a file header over here, and then we are minimizing the header, and then we're converting the sequences itself and the basic quality all together into a new format. So the first thing that makes sense to all of us is to remove anything repeated. Those are the header line, the header format, and those are the technologies. And we will look deeply at those header line. You'll see always the instrument is recorded in every record that belongs to read. Again, the file could contain like millions of reads, and the instrument name is repeated in all those reads. So you just get it once, store it somewhere at the beginning of the file, done. Then you are minimizing all the header in that FASTQ file. And you can do the same thing for run, flow cell, and so on. So we'll identify all those variables that are common between all the reads in a black color. So those variables, you can safely isolate them and put them in the header of the file. And things in red, you cannot optimize them further because they are really unique to each read. So you can keep them in the header file or in the header line for each read. There are things like blue. This correspond to the paired end reads. So these information, especially in Illumina technology, will tell you if this read is coming from the left side or the right side of the genomic sample when the sequencing machine starts reading it. And this is how it looks like for our file header. So if you open our file format, FASTA, this is what you'll see in the top of the lines. And basically, it's related to those variables highlighted in black color. Okay, so anything that is not, or it's common basically between the reads, so we move it here. For example, the instrument ID, the accession number, there is no point to repeat it in all reads. So you can just record it once over here. But also we record things related to how we get our file format. For example, the mode, this is something related to our tool. So there are 4 modes to process the file. So we record it over here so that if you want to convert back this file into FASTQ, you don't need to provide any parameter. So it will read it directly from the header and then convert everything. And more important thing, we are calculating ourselves the quality that's provided in the FASTQ file and provide you an estimate of the equation that might be used to scale those qualities. And I'll tell you why we are scaling those quality, but this is extremely important. We are telling you in advance what you expect as a range of qualities. And this is extremely important for quality control to filter the data easily and safely, rather than just trying to guess every time by reading read by read. All right, now let's move from the file header into how we change the base and the basic quality into something else. Our goal is to replace, remember, those bases, the ACGT data, we have it, each of them stored as a character. Character mean 1 byte, so 8 bits. And there is the basic quality, another 1 byte. So this is 2 bytes per character, which is the ACGT chemical molecules. We want to encode both of them in 1 byte instead of 2 bytes. Okay. Now, how to do that? There is a naive solution. We have 1 byte. So what we can do is split it into 3 bits and 5 bits. The 3 bits are safe to encode A, C, G, T. And we need a 5th character, which is the N, because sometimes the sequencing machine cannot tell you if this is A or C or G or T. It will tell you something else, which is N, which is unbiased or unknown character. Okay. So we need 5 bases to encode them, and the minimum number of bits to achieve that is 3 bits. So this is a naive way to do it, right? And what we have remaining is 5 bits. Those 5 bits are enough to encode 32 basic quality values. Okay. So if this is like very low quality of A, so we record it here 000, it means A, and then we record 0000, it means very low quality, and so on. However, we are proposing something else. We said, okay, the 8 bits could represent 256 values, right? We agree on that. Now, we said, let's implicitly encode the ACGT data. We don't want to record it, okay? And we want to split that range. This is from 0 to 256 values, right? We want to implicitly divide that range into sub-ranges. Now, the first one, N, which is the unknown character from the sequencing machine, most of the sequencing technologies report it as single quality value all the time. It doesn't matter. It will not tell you, oh, yeah, it's unknown, but low quality or high quality or medium range. So we said, OK, let's give it just 3 bits. It's really not needed. We can have it with 1 bit, which, for example, PacBio always with exclamation mark. So always they have that quality assigned to the N. But for our case, we have it as extra bits. So we just give it like 3 values. Okay, so it can be 0 or 1 or 2. If you see 0, 1, or 2 recorded, it means it's N character. And we said the 0 is low quality, the 2 high quality, and so on, in case of any sequencing technology would need that. Now, the remaining values, we divide them equally into 62 values per character. So if you see 3, it means it's A with low quality. If you see 65, it means A with high quality, and so on. Likewise with the C, so if you see 66 or 128, that represent the C, but with different qualities. Likewise with the T or U, T is needed for the DNA, U is needed for the RNA. So you could encode DNA and RNA using this diagram. Now we only have 1 remaining value, which is 255. I'm going to talk about it in the next few slides. We'll see that. Okay. So this is really how we encode the base and the basic quality all together in 1 byte. And this is faster to decode. You really don't need to split the 3 values or do any kind of multiplication with this value in order to isolate the 3-bit alone and then figure out what kind of ACGT you are having. So just by looking at the value, you already know what base and what base equality you are using. And apparently, you see wider range for the quality score. Here, we can encode only 32 values, but over here, we can have 62 values for equalities. And this is really wide enough to cover all sequencing technology. In Illumina, we need only about sometimes 3 or 4 values, sometimes 40 if we are using a very old data. And likewise with PacBio. In ONT only, we might have some issue with the very old data. They have extremely wide range, about 100 values to represent the equalities uniquely. Okay, so by having the 62 values, you could represent the higher values, and the low value you can scale them back to the lowest value and something like that. But when we talk about this very old data, it's from 2006, which has like 60% of equality, okay, which is useless these days. Now, the other issue we came through, since we combined the base and base equality together, then we are going back to the whole spectrum of ASCII character from 0 to 256. And by doing so, you expect to have a new line character in the middle of the line. So imagine this is of value 92, which means something like C or something like 92 would be here, right? C with middle quality. 110 will also be in C with middle quality or a little bit high quality. And this is expected. You will have these cases. Now, if you have a new line in the middle of the line itself, that will cause a lot of issue with interpretation. So you will be confused with this line, with this character and this character, right? So to solve this issue, we said we already have an extra value over here. So if we know it's the end of the line, just replace it with 255, okay? We don't have the new line character recorded. So in this case, we just rely on this value. Whenever we came through 255, that's the end of the line. Go to the next line to the processing, right?
It's a binary sign.
Yes, it's binary, but depends on how you interpret it. So you cannot use the FASTQ parser to interpret those files, and you need to find a way to parse those files. So we try to provide the API in order to parse these files. And one of the things that we should look for is, for example, the 255 in binary, of course. So this is byte value, 8-bit value. Now, if you have this originally from what you got from the sequencing machine, or the SRA, sorry, because you can see the accession number over here, this is how we convert it into our file format. So this file header is only recorded once. So if we have 5 lines or 5 reads over here, we will see only 10 lines after the header, because 2 lines correspond to 1 read. So we record the header, but remember, we optimize it. So we remove the accession number and many of the details that we move here. And this is what we got from the header. And then here, what we got to combine the base and base equality together. Now let's go back to the results. Okay. This is the FASTQ file, the original file. This is what we got from our file format. So the minimum that you should expect is about 2.6x. The 2x improvement is by combining the base and base equality. It makes sense, right? And the 0.6 is from optimizing the header. Okay. However, sometimes you really don't need the header at all, especially if you rely on serial number just to tag the reads, like from 1, 2, 3, 4, 5. You can do that while you are processing the read. You just read one line that corresponds to the read itself, and then you can assign it any number that you like. Okay. That number could be unique internally within your software, but doesn't need to be globally applied to the whole genomic analysis. So if you do that, you will achieve something like 3.15. But this is really very aggressive optimization. So this is what we expect all the time. Okay. Now the file format is fully reversible. You can convert it back to FASTQ. There's nothing that you'll lose. You don't need any kind of decompression to read it. So you just read the values in binary and you convert them directly into bases and base equality. You can do that by lookup table, very simple lookup table. And we already did that in Minimap too. So we already change Minimap2 slightly in order to be compatible with our file format. Now, if you are familiar with genomic data, we always use gzip to compress it, right? So you could compress our file, could compress the FASTQ file. Let's see the comparison. So if we compress the FASTQ file, and if we compress our file, we will see much faster compression time. Why? The file size is smaller. Makes sense, right? You have 2 point something smaller file. If you compress it, it's much faster than the larger file. But also it will give you a benefit in terms of decompression, which is extremely important for the analysis tool, because the analysis turns out to not to decompress the file in advance. So they don't decompress the file first and then they start processing, but rather they have the gzip as input directly, and they do the decompression on the fly. So it will cost them about 2x slower performance compared to having a file that's not compressed as input. So it's really a big issue for the analysis file. And when we have faster decompression, that means you have faster analysis later on. Now, because we reduce the data movement, so think about everything we say in the beginning about all this motivation, how you move the data around, you move it from one component to another. Now, by having less data, you expect to have a lot of benefits in terms of everything, memory, energy, and all these metrics. We just released the paper a week ago. We submitted to ISMB. Let's see the results. So this is a QR code. This is a collaboration between Belkent, University of Southern California, and our university. And the main lead behind the implementation is Adrian. And the funny story about him, he's a high school student. He did great work. He's been with us since 6 months. He's extremely good in implementation. Okay, so we trained him for bioinformatics. We give him the idea. Then he did a really great job. So you'll see his name in GitHub. He's keep pushing since we released the code. All right, now let's go quickly into another topic still related to the data. I'm not sure how many minutes do I have. Okay, I'll try to finish in 10 minutes and then we can take questions. All right, now how we could minimize data by doing what we call data sparsification. We are still focusing on the data for the whole talk. So we'll show you different use cases, how we could improve the data or how we could improve genomic analyses by just improving the data itself. Probably when you get sick, you go to the hospital, you try to ask the doctor, okay, I have fever, I have diarrhea, I have many other symptoms. What should be the medication? And the first thing they will do, they will take a sample, they put it in a plate, they keep it for 1 to 7 days until it grows. And then based on the shape and the color and many other things, they could identify what bacteria, what virus you have. And this is the traditional method in almost all hospitals, okay? However, this is taking a lot of time. So you could be already cured by the 7 days, right? Or you could be suffering by trying different antibiotic and you're not getting better. But also, not all species are culturable. So you cannot do this method for all kinds of viruses and bacteria. And we are replacing this traditional method using what we call metagenomic analysis. This is somehow a new field, starting like 15 years ago, and relatively new compared to the genomic field. What we do, we collect those samples, we send them to the sequencing machine, we generate what we call FASTQ file, and then instead of using single genome, if you remember this picture, instead of using single picture as a reference, we are now using the whole database of all known species to compare it to the FASTQ file. Okay? But this is extremely expensive process because the database here is about 4 terabytes of data. This is RefSeq database. This is the most comprehensive database around the world. Okay? So imagine every time you get a FASTQ file, compare it with 4 terabytes of data in order to identify what virus or bacteria. So we said, okay, we want to fundamentally solve this problem. Imagine you have a reference sequence. Imagine you have a read. Now, if they are exactly the same copy, you'll get exact match directly, right? But now we want to reduce that amount of data. We said, what is the minimum number of bases that you can use to match them without using the whole sequence? One solution could be just to take every other character. Really take half the number. You don't need the whole sequence. Okay, just take half the number of characters. We want to take every other character. So take one, ignore one, take one, ignore one, and this is what you need to process, okay? Or what you need to store. Now, this is exact match because originally they are exact match. So it doesn't matter. As long as you apply the same pattern to both of them, you still maintain that. However, real life is not as easy as that. Remember, this is a reference genome, but this is a read. A read means it's randomly chopped from your DNA. So you randomly break down the DNA into smaller pieces. And you have no guarantees that you all the time you read it from the first character. You might start from second, third, fourth, and so on. You might any point start reading that read sequence. Now, imagine if we just start the read from the second character. This is what you get from the sequencing machine. Now, if we want to apply our pattern, take one, ignore one, take one, ignore one, this is what you will get. Now, if you try to match them, they are exactly the same, but we just like ignore one character. Because the sequencing machine is random. Again, it can read your DNA at any point. No match at all. No match at all, although they are the same. Okay. Now, how to solve that issue? Let's get back to our copy. So we have the same copy. Now we want to apply the same pattern to this sequence, but we don't want to apply it from the first character. We said, okay, what is the pattern used here? It's 1, 0, right? And then repeat it itself. 1, 0, 1, 0, 1, 0, 1, 0, 1, 0, right? So the minimum pattern, minimum repeating pattern is 1, 0. So it's 2 character. So we have 2 options in this case because the pattern is 2 or of size 2. Either we apply that pattern from the first character or we apply it from the second character. And now we need to take in consideration both cases. So if we apply the pattern from the first character, this is what we'll get. If we apply it from the second character, this is what we'll get. Try to match only a few characters. Don't apply the pattern to the whole thing. Just try to apply it to a few characters. And let's see which one of them exists in this sample. Okay. Now we see that this one exists. So it looks like this one is better mechanism to apply the pattern. So we choose this over that. And then we apply it to the whole sequence. And then we use this for matching. And then we get the exact match again. So this is a simplified method to what we propose. We call it genome on diet. And this is a way to handle a huge amount of data. We cannot use this method for genomics, although we show some benefits, but I don't recommend it. We recommend using this method because we are specifying the data, so we are losing half of it. We recommend to use this for only huge amount of data, okay? Now, there is a previous concept called space seeding. So we got a lot of question how your method is different from space seeding. In space seeding, what we got, we have the genome, we extract what we call k-mers, and then we apply the pattern to the k-mers, okay? So the resulting k-mers will be this shape, okay, after applying the pattern. What we do in genome on diet, we are applying the pattern to the genome itself, not to the k-mer. How's that different? This is what we get as a result after applying 1, 0, 0, 1, 0, 1. This is the minimum, or sorry, this is the minimum repeating pattern. We apply 3 times over here. So this is what you get as a result. Now, these are the resulting k-mers, and you can see how different they are from the space seed. Now, what benefits do we expect out of this? We expect a lot of benefits. The most important one is the reduced execution time, because imagine we convert this whole sequence. This could be a genome, by the way, not necessarily a read. So when you think about like 3 gigabytes of data, like in a human genome, and then you got out of it only 1/3 or so. Now, where this method could be helpful, this method could be helpful when you are comparing like relatively long sequences together, and you know they would match at some point, right? But you need to know where they match first, and then you do fine-grained analysis, okay? So you use it to reduce the amount of data. Instead of searching in very long sequences, you search only in that. And if you know in advance they exactly match, like when you look for viruses or bacteria, then you would get a lot of benefit of that because you use minimum amount of data to match them. Not only that, in viruses, they are mutating themselves very frequently. So they introduce their own mutation every 2-3 months. So although you know the virus is this genome and so on, but after 2-3 months, they start changing themselves. They introduce their own mutation. And by allowing for spaced character over here, you are allowing for those mutations to be detected, okay? Because you are not dictating the exact match to be at specific location. We already compared for 3 applications, but the most important one is the metagenomic profiling, and we got about like an order of magnitude faster and 2 order of magnitude less storage requirement to store the 4 terabyte of data from the database itself. We have a lot of results to compare Minimap 2 and other application and how we can improve that. We got the paper published in Nature Communication last year, and we have the source code already publicly available, so feel free to try it. Maybe I'll cover this quickly in 2 slides, and then we can take questions. So now, how to minimize data movement by filtering the data earlier. We know that, hopefully by now, that we have the sequencing machine. We generate a huge amount of data. Then we start doing the computational steps. One of the computational steps is to convert this electrical signal into reads. And then we do quality control. Then we do read mapping, then assembly, then many more analyses. Now, this data that we got from the sequencing machine, sometimes of low quality. Sometimes it's really useless. We don't need it. But we figure out that at very later stage. So we generate the whole data, sometimes like 60 terabytes of data from the ONT. It's extremely huge file we got from there because raw signal value. And we do the base calling, we do the quality control, and then, 50% of the file is low quality. Let's remove it. So it's already too late because we already spent a lot of time doing computation. Base calling is something really expensive for ONT data because it's based on machine learning. So it's not like direct mapping from one value to value. It's not like that. So we have to really guess it and figure out what's the output. Now let's see this. Hopefully it will give you the idea quickly. This is traditionally how we do it. We start base calling, base calling, base calling, base calling until we finish the entire data, and then quality control, and then read mapping. What we are proposing here is to overlap all these processes. So the moment you start base calling, and it will take some time, actually. You have about 12 hours to do this, the base calling for about 24 hours of sequencing. So it's almost half time of sequencing. If you spend like 1 day doing sequencing, you expect like 12 hours doing base calling. Now, as you do base calling, if you got a chunk of data, then this is ready for doing quality control. Do quality control on that ready data directly. And then you can do whatever you want next computation on that quality control data. You can do the same for next, next, next. By the time you finish base calling over here, you'll have just little overhead to complete the whole process. So the solution is very simple over here. But when we talk about the grand scale of the data, it's great benefit we achieve by that. And we optimize what we got over here because the moment you reduce the data for read mapping, now read mapping becoming faster and so on. So this is the speed up we got out of that compared to the CPU and GPU results. And this is the paper we call GenPet. That's already publicly available with the source code. Now we have something more, but I'm going to skip this work. This is about how we do filtering for data-aware filtering. It means when you know the data, for example, it's Illumina. Illumina is well known that 80% of the FASTQ file that you are getting is exactly matching the genome. So 80% of the FASTQ file you are getting exactly matching the genome. So there is no point to do read mapping using very expensive algorithm like sequence alignment, because you already know in advance that 80% exactly match the genome. So you could use like Hamming distance for the entire file until you figure out those 80%, and then you apply sequence alignment only to the remaining 20%. So this is the whole idea of this work. I'm going to skip it. keep more time for discussion. But this is what we call as Gen Store. It's already published out there. Now, I will end up with this conclusion from Illumina. Their post on LinkedIn, I think 4 days ago or something. They said the industry generates upward of 40 billion gigabytes of genomic data every year. Getting the data is no longer the problem. What is the problem then? Making sense of it. And we really need to spend time on how to optimize the data itself and the analysis rather than on the sequencing. Sequencing these days is really much better than before. And we can do high throughput sequencing. For example, we can generate data for 48 samples at the same time. And this is what my group tried to focus on, the whole compute stack from genomic data to algorithm to software to the hardware. We focus on all kinds of genomic analyses, not necessarily genomics or read mapping, but we focus on metagenomic and pangenomics and many more. I'd like to acknowledge AMD and AMD HPC for their generous funding, and state of Georgia and our department for the startup fund. We are teaching a lot of courses. All of those material are online. We can see advanced bioinformatics, for example. The advanced computer architecture courses are all public in our YouTube channel. If you ask me to recommend one single paper to read to get more ideas about this paper, I would recommend this extensive survey paper. So if you go through it, you'll see a lot of references, a lot of things covering even the sequencing step itself, the chemistry behind how we do the sequencing itself. All right, that's the end of the talk. Thank you so much. Yeah.
Thank you, that was amazing talk.
Yeah.
With the FASTA, like obviously all the software nowadays is out for FASTQ, so it's completely what design FASTA, which is fine for the right solution. I would say at the moment the three biggest issues in clinical with these files would be the BCL file. So if we're in 48 patients, we have 48 patients in that one file. If one patient then decides I want to opt out, it's fine. We can remove their FASTQ files, bad files, but we can't remove the BCL file, which contains all the patients. Second would be the processing. Obviously, it takes a long time. We need to take control. And the third part would be the BAM part of the system. So we'd want a solution that fits all of those problems. So like yours would definitely increase the speed, but then with the other two, would you see a solution?
Excellent point. I like that. So for the PCR, normally we have like unique tag, right, for each patient or for each genome. So the easiest solution would be to split these files into, if you say like 40 patients, then you split them into 40 files, convert each file into FASTA format. Okay?
What we do in like the multi stage, but I think we usually like keep the file that straight off pizza in case any mistake that's made further down the line.
That's true, but don't use it for analysis, right? You keep it some time to go back to it, right? But the problem that motivates us is the analysis part. So you can keep that safely somewhere. You can compress it, keep it untouched. But to go for the analysis, you could go with FASTA, for example. And you'll never expect any loss in the data because we don't remove any kind of piece of data from the file itself. So we maintain everything as is. Only the interpretation is different now. Instead of reading ACGT and basic quality, you need to read values, and those values just convert them into two of these. And we already show a proof of concept how you can do that in Minimap2. If you are aware of read mapping, Minimap2 is the most widely used one. So we already show how we can change that, tweak like just small lookup table, looking up those values into the bases without affecting the SAM accuracy or the BAM and so on. Now for BAM, it's very expensive to build, right? You need to do read mapping to build the BAM file. Okay? You can do SAM from the unaligned data without aligning to the genome, and that will be less expensive. You'll not get compression as if you get from the BAM file. So we have a comparison with BAM, with CRAM, with SAM, with all those existing file formats, and you'll see a huge difference with our file formats, and we get the same compression rate. Yeah, my pleasure.
I would like to know, if possible, what is the difference between
the compressed file sizes within FASTA, because you have to the size, but you have compressed.
That's a very good point. I think we quantify that in the paper. I cannot recall the numbers, but it's much smaller, much smaller. Now, there is a trick in the compression because there are like 12 levels of compression. So it's up to you to use from 0 to 11 in gzip, for example. So it depends on that, because the more you compress, the more aggressive you go, the less repeating pattern you'll start seeing, and you'll start losing that benefit. Okay, so if you use like 0 to 6, for example, you see the huge benefit, which is the default of the gzip. But once you move to more compression level, you'll not see any difference in the compression size, especially with like the biggest. But you'll still see benefits in compression time and decompression time. Okay, yeah.
So my question also goes along those lines. For what it seems, what you're proposing is a reformatting the FASTA, but you are not applying the entropy compression to that directly. So mostly compressors rely on the redundant of the file. Have you explored anything both methods?
That's an excellent point. I would say we are enriching the repeating pattern by having a lot of zeros on the binary data. If you open our data, you'll see normally it's similar to one-hot encoding, for example, because once you merge the bases with the base equality, you'll see less number of ones, but you see a lot of zeros along that path. Now, why we have more zeros, it really depends on the data. You could have a lot of C's, a lot of A's, a lot of low quality data, and that will affect the repeating ones and zeros in there. Now, we don't want to apply the compression because this is not our goal. Our goal really to organize the mess that we have in the FASTQ file. But we have a lot of great existing compression that you can use, whatever, like gzip, one example. We have XZ, we have many other variation of, so you can use it. And it's independent of our work. And we prove a lot of benefits by applying those existing compression tool on our file format versus the FASTQ. Now, there is a great point why not to develop dedicated compression tool for our file format. And we see this benefit with FASTQ file. So there is a lot of specialized compression tool for FASTQ file. They are not general purpose like gzip. You can compress any type of text. And you'll see more benefits if you do that. So this could be the next step, is to build our own compression for the FASTA or faster file. But that's a great point, like how you could, instead of converting it into a sequence of values, you could find something else. But then it will be compression, it will be hard to decompress, and then we need to account for that. That's a great point.
Okay, a follow-up from me. If you were to develop your own present team, would you operate on compressed data?
It depends on how aggressive you go with the compression.
Yeah, but then there is a co-design there.
That's true.
If you are going to do your own.
I like that. I like that. But it really depends on the compression that you're doing. You might spend more time on the decompression, and that will be accounted in the analysis, especially if you want to do it on the fly. Like you don't want to decompress first and then start the analysis. But that's a great point, and most of the tool are doing it. Most of the tool accept gzip file, then they start processing without the requirement of unzip that file. But you'll see the execution time is going up.
But you don't, if you depend on how you compress, you may not even have DMZ on the fly. You could have to compute on the.
That's true. That's true. That's true. But again, depends on the compression algorithm you are using. There is always trade-off. Yeah, there are a new work on how to accelerate the decompression, for example, on the fly, or computing directly on the compressed data without the need for decompression. So we could merge these work together. But that will be something fantastic to have.
Another question?
Thank you so much, Adrian. Thank you so much for coming.